I've added the clicker questions to the curriculum here because I often will do these in class, which I find is a very good tool to give students a baseline of how well of a grasp they have on the particular material. So what I'll try and do is on the incorrect questions, or if they're true or false, or if there's a multitude of different questions on multiple choice, I'll try and go through each one and explain why which one is correct and which ones are inaccurate. Number one, DNA polymerases in eukaryotes are used to A, repair DNA after replication, B, replicate DNA, C, repair DNA after damage, or D, replicate telomeres. As you can plainly see, this is clearly E, all the above. The DNA polymerases, all different types, will do all four of these particular things. Two, what role does primase play in DNA replication? A, primase initiates DNA replication of leading strand followed by allowing DNA polymerases to bind. Or B, primase synthesizes the RNA primer needed to initiate synthesis of Okazaki fragments. Or C, primase removes the short RNA segments produced during lagging strand replication. Or D, primase acts as an exonuclease to hydrolyze DNA in 3' to 5' direction. The answer is B. Primase synthesizes the RNA primer needed to initiate synthesis of Okazaki fragments. Very simply, the polymerase is what takes care of the leading strand, and very simply, it's going to be the other primase is going to remove the short RNA fragments during the lagging strand. As we know, it's going to go in the 5' to 3' direction, so letter D will be inaccurate as well. 3. Okazaki fragments are a consequence of A, the need to unwind the DNA molecule during replication, B, the circular nature of the prokaryotic chromosome, C, the presence of histones in the eukaryotic chromosome, or D, the ability of DNA polymerase to synthesize only in the 5' to 3' direction. The answer is D, the ability of DNA polymerase to synthesize only in the 5' to 3' direction. The other answers shown here, as we can see, the Okazaki fragments have nothing to do with the unwinding of the DNA molecule for replication. They're not present in the prokaryotic chromosome because it's a circular chromosome, so simply goes in one direction for replication, and it does not have anything to do with histone capability in the eukaryotic chromosome. 4. What prevents RNA nucleotides from becoming incorporated into a growing DNA strand? DNA polymerase discriminates against nucleotides containing the ribose sugar. Or B, complementary base pairing means the RNA nucleotides cannot bind to DNA nucleotides. Or C, DNA is synthesized in the nucleus, whereas RNA is synthesized in the cytoplasm. Or D, RNA synthesis is halted during the S phase of mitosis. The answer is A, DNA polymerase discriminates against nucleotides containing the ribose sugar. This is going to help prevent the RNA molecules from remaining bound to the growing DNA strand. The answers here, B, C, and D, have nothing to do with the growing DNA strand. Number 5, true false. DNA replication moves in both directions in prokaryotes and eukaryotes. This is true. So of course, as you know, in prokaryotes, we have a circular DNA strand, which is going to go simply in one direction. And in eukaryotes, we're going to have the different replication forks, often in the groups of thousands, all at the same time. They go in both directions as regards to the replication fork, but of course always going in the 5' and 3' direction. So you have both the leading and lagging strand off each portion of the replication fork. Number 6, telomerases are necessary in eukaryotes because, letter A, eukaryotic chromosomes contain histones, which must be replicated with DNA. B, telomerases are the only eukaryotic enzymes that can replicate DNA. C, eukaryotic telomeres are necessary to preserve the integrity of the kinetochore. Or D, eukaryotic DNA polymerases cannot copy all the way to the end of the chromosome. The answer is D, eukaryotic DNA polymerases cannot copy all the way to the end of the chromosome. As we talked about before, the 5' ends cannot be synthesized by DNA polymerases, so telomerases are going to be special enzymes that are going to be a reverse transcriptase that can help extend the 3' end overhang on the 5' complementary strand. As far as the other answers here, the eukaryotic chromosomes contain histones. That does nothing to do with the telomerases. Telomerases, for letter B, are not the only enzymes that help replicate DNA, as we've talked about before. And of course, the kinetochore being shown at the middle of the sister chromatids is more importantly used with the mitotic spindle during mitosis. And exactly the opposite end, you find the telomerases, which are only found at the ends of the DNA molecule. Number 7, what is the main difference between base excision repair and nucleotide excision repair? A, base excision removes one nucleotide. Nucleotide excision replaces an oligonucleotide. Or B, base excision involves one catalytic step. And nucleotide excision has multiple enzymatic steps. Or C, base excision can repair different types of damage, whereas nucleotide excision only repairs a specific type. Or letter D, base excision involves DNA polymerase and ligase. Nucleotide excision uses DNA glycosylase. The answer is A. Base excision removes one nucleotide, and nucleotide excision replaces an oligonucleotide. So the best example I can give you on this one is the base excision is going to remove one particular base nucleotide, such as uracil, which should be replaced by a thymine, whereas a nucleotide excision is going to replace a larger strand of bases known as oligonucleotide that could be damaged from either UV light or possibly from some kind of carcinogen. As far as the other answers here, will go pretty much as like a true-false statement for each one. The base excision involves one catalytic step. We know that's false because it's going to involve a number of different steps with different types of enzymes, with DNA glycosylase, and AP endonuclease, and also a DNA polymerase, and finally a ligase. Letter C, base excision can repair different types of damage. This is pretty much just basic repair of one nucleotide, where the excision only repairs a specific type. Could be any type of damage to the DNA, so it could be from multiple sources of damage. And finally, letter D, base excision involves DNA polymerase and ligase, whereas nucleotide excision uses a DNA glycosylase. Basically, they're going to use both different types of DNA polymerases and ligases, whereas the nucleotide excision is not going to use the DNA glycosylase. It's only used for base excision. Number 8, defaults in mismatched repair mechanisms affect only genes that regulate cell proliferation, thus leading to colon cancer. Now, this answer is false. Very simply, any gene can be the subject of a bad mismatched repair mechanism. So this can lead, or actually be led from any particular cell line. It could be a liver cell, it could be a colon cell, it could be a muscle cell, any number of different cells that could be affected by this particular gene. It doesn't necessarily have to be from cell proliferation. It could also be from the transcription and translation of different types of proteins as well. So unfortunately, this type of mismatch repair mechanism can affect any number of different genes. Number 9, how does translesion synthesis differ from excision repair? Letter A, translesion enzymes skip over mismatched bases. Excision acts directly on them. Letter B, translesion enzymes work in the 5' 3' direction. Excision enzymes work in 3' to 5' direction. Letter C, excision repair acts before DNA replication. Translation synthesis acts after DNA replication. Letter D, excision repair replaces single base pairs. Translation synthesis replaces large sections of DNA. Now, this is a twofold answer. This comes from your textbook. Generally, when I do these, if I'm going to put multiple answers in here, I'll actually give you a selection A and C, or B and D, or simply all the above, or none of the above. But in this answer here, both translesion enzymes skip over mismatched bases, excision acts directly on them, and letter C, excision repair acts before DNA replication, and translation synthesis acts after DNA replication. And we follow through with the true-false statements for letter B and D. Letter B very simply is false, since excision enzymes do not work in 3' to 5', they work in 5' to 3'. And finally, excision repair replaces single base pairs. That is incorrect. It can actually take care of large swaths of the DNA, and translation synthesis actually will take care of very small pieces, usually the dimers we had discussed in the previous chapter. So both B and D would be both false. Number 10, recombination repair of double-stranded breaks can introduce A, base substitutions, B, base inversions, C, base deletions, or D, base duplications. The answer is C, base deletions. Now, what's interesting about this particular question is it neglects to put in base insertions as well, which is also a possibility with recombination repair. Very simply, if an immediate ligation is not possible of the double-strand break, there are going to be different types of nucleases like Artemis, which will remove the nucleotide, or the DNA polymerases, sigma and gamma, which can add nucleotides before ligation of the LIG4. So you can get deletions or insertions respectively. but simply the base substitutions, inversions, and duplications are simply not possible with the double-strand breaks. Number 11, homologous recombination repair requires the presence of a centromere because, letter A, the repair requires using information on the undamaged sister chromatid, letter B, the repair relies on proteins associated with the kinetochore, or C, the DNA polymerases used in repair are only expressed during mitosis. The answer is A, the repair requires using information on the undamaged sister chromatid. So this is textbook for the homologous recombination using the homologous area of the sister chromatid that is therefore undamaged as a template. As far as the other answers go, the repair relies on proteins is not necessarily associated with kinetochore, and of course, DNA polymerases used in repair are not only expressed during mitosis. Not quite a true-false question, but actually more of a very minute multiple-choice question, number 12. Does gene rearrangement happen before or after transcription? The answer is A, before transcription. Gene rearrangement is the primary mechanism that we see the diversity in different types of immunoglobulins, both the light and the heavy chains. So the germline DNA will rearrange different DNA in different types of lymphocytes to basically provide a primary transcript to allow the immunoglobulin mRNA to be translated into the appropriate antibody. Number 13, antibody diversity relies on both gene arrangement and A, post-translational covalent modifications, B, post-translational RNA splicing, C, pre-translational covalent modifications, or D, post-transcriptional RNA splicing. The answer is D, post-transcriptional RNA splicing. As we look at the answers here, the letter D, post-transcriptional RNA splicing, you should think about the order in which the RNA is going to be modified and also translated. Very simply, the other answer doesn't make any sense. The covalent modifications wouldn't really change the actual amino acid chain. So post-translational covalent or RNA splicing, so that simply doesn't happen, would not be at play here. And the pre-translational covalent modifications we also know would not be happening as well. But definitely the post-transcriptional, so after the transcription of the actual mRNA, the splicing of the introns and recombination of the different regions will lead to the different types of antibodies that we see coming off the B lymphocyte. 14, true or false? Cytosine deamination is a beneficial process. This is true. Cytosine deamination converts cytosine to uracil and contributes to antibody diversity. And the final question for this chapter, number 15, gene amplification would typically be found only in A, aging tissues, B, mature tissues, C, damaged tissues, or D, developing tissues. The answer is D, developing tissues. Now, typically see more gene amplification with increased numbers of replication. So naturally developing tissues, as they're constantly mitotically dividing, you see more gene amplification. Although you could make the argument that damaged tissues, in particular if you're trying to restore, let's say, epithelial tissue or muscle tissue or even different types of nervous tissue, which some can replicate, not many. But you could say gene amplification could also be found in damaged tissues, but mostly found in developing tissues. Conversely, aging tissues or mature tissues typically are not going to have as much gene replication. So typically, again, if you read the question, it's not perfectly worded, but I think it might word a little bit differently. But again, typically you find gene amplification found only in developing tissues.